in this session, our panel will focus on innovative approaches for presentation of clinical safety data into tables and figures, while touching on key standard safety topics introduced in the clinical guide presentation. So our panelists for this session are three, and I would invite them all to switch on their video so we can have a discussion. And if we can go off the slides, would be great. So our panelists for these sessions are Mary Nielsen, who is the Executive Director of Statistics in the Safety Analytics Group at Eli Lilly and co-lead of the FHUSE Safety Analytics Working Group. Bess Leroy, who is the Head of Standards Development at Clinical Data Interchange Standards Consortium, or CDISC, and Jeremy Wildfire, who is a data scientist at Gilead and technical lead at DIA ASA Interdisciplinary Safety Evaluation Working Group Interactive Safety Graphics Subteam. So to begin, each of our panelists will have 5 minutes for their opening remarks, and we will start with Mary. Thank you.
Hi. Yes, thank you for including me in this panel. I'm happy to share a lot of activities going on within an FDA FHUSE collaboration. So thank you for allowing this opportunity. We have a lot of work we can share with you as it relates to standard safety tables and figures. Next slide. So I'll provide a quick background of the FDA FHUSE collaboration, and I'll summarize some of our final deliverables and summarize our ongoing projects and talk about next steps. Next slide. The FDA approached FHUSE in 2012 to begin a collaboration. And the idea behind this collaboration was to provide a platform for academia, regulators, industry, technology providers to work together to solve some very important gaps and needs and common challenges. And this collaboration is supported by FHUSE, CDER, and CBER. This particular collaboration currently hosts 7 working groups. Safety analytics is one of them. The others are data transparency, data visualization, emerging trends and technologies. There's a non-clinical group. There's a very active optimizing the use of data standards group and a risk-based monitoring group. And then safety analytics is the 7th working group. And within each of these working groups, there are several multiple projects going on within each of them. In fact, over the past 10 years, there probably have been well over 50 projects. So there's been all sorts of good work and activity going within this collaboration. So if you're not familiar with it, I do encourage you to get familiar with it. If you go to fuse.global, click on working groups, you can see a summary of the working groups and projects that are occurring today. And then there's a deliverables button in which you can see all the deliverables that have been published since the collaboration started. All the FDA FHUSE collaboration deliverables are publicly available. You don't need to be a FHUSE member in order to participate in a project. It's also open. You don't need to be a FHUSE member. So it's a very open, transparent collaboration. Next slide. So with respect to standard safety tables and figures, we have had over the last 10 years, multiple project teams who have worked on standard safety tables and figures. And these project teams have come primarily from 2 working groups. One is the Standard Analyses and Code Sharing Working Group that existed from 2012 to 2020. And then our Safety Analytics Working Group started in June 2020. And I did include the description of the Safety Analytics Working Group. We are a cross-disciplinary group, so primarily medical, stats, data sciences. And the main goal is to improve the content and implementation of clinical trial safety analyses. So we share the passion for standardization. We think standardization is very important to improve implementation. And when standardization is done correctly, it can also improve the content of our analyses. But also to improve the content of analyses, we feel education is a big part of that. So we have a big educational component to our work. A lot of our deliverables include instructional and rationale and references to education. We also have had some, recently a educational module added to the FHUSE education section of the page. So education is a big part of what we do in order to improve the content of safety analyses. Next slide. So here's an example of our FHUSE deliverables that relate to standard safety tables and figures. So we have had, starting in 2013 and 15, deliverables related to standard analyses and displays for labs and vital signs and ECGs. We had an adverse event white paper. You may have heard or saw in the integrated guide mention of a study size adjusted percentage. We're big advocates of stratifying analyses and providing appropriately stratified summary statistics when warranted. And so this video you can consider very supplementary to what you have seen in the guide and Matt's talk. Similarly, the adverse event white paper does have mock tables and figures, but it also has a lot of educational components as well and very important references. We have one on demographics, disposition, medications. We have had work related to an interactive display in a submission, right? An interactive volcano plot for adverse event data. Greg already alluded to this to some extent. So we do have experience now providing a display with interactive features for adverse event data. We have work related to adverse event collection and treatment emergent definition. There's a safety topics of interest white paper that can be considered a carry-on topic to general adverse event displays and data listings, and then another updated lab. So all of this can be considered supplementary. The main audience for the analysis and displays white papers are industry. So it's mostly industry clinicians, statisticians, and data sciences. And it also includes for individual studies and for integrated summaries of safety. So the FDA integrated guide, the main audience is clinical reviewers, and then the FDA programmers who are providing displays for the clinical reviewers. However, you would think that what we produce in our submissions, what industry would create and what clinical reviewers would want, you could consider that, thinking that should be very similar and not identical. So we'll definitely be working together on updating some of these white papers. Next slide. To find these deliverables, you can go to FHUSE.global, click on the deliverables button. Hopefully that's very easy. Next slide. We have several ongoing projects, and a lot of these do overlap with the FDA's integrated guide. We do have a group on liver analyses, and we still have a group on lab analyses. We have an adverse collection group, treatment emergent definition. We have a whole project team devoted to treatment emergent, safety analytics. Greg already mentioned the adverse event groupings and safety. That's a new project, and so that's certainly could be considered supplementary to the FMQ effort. And then we do plan on initiating a project to gather comments on the FDA docket. Next slide. So next steps, we will continue discussions at next week's FHUSE Computational Science Symposium. We do have a breakout session devoted to gathering some initial feedback, and we will have a project team to gather feedback as well, and we'll target October 31st to get that together. And that will include a comparison with existing FHUSE white papers. And we will over time discuss plans to potentially updating some of our deliverables to incorporate some of the new thinking on these displays. So that's all I have, and thank you for the opportunity to share this.
Thank you, Mary. Now we will hear from Bess Leroy.
Thank you, Vaishali. So again, I'm Bess Leroy. I'm from CDISC, and I'm the head of standards development. Next slide, please. So I want to just provide a little bit of background, in particular on CDISC's perspective. So I think, obviously, like the team has shown here, we've seen unnecessary variation in analysis results reporting. And to date, we have limited CDISC standards to support analysis results and that associated metadata. Over the last few years, CDISC has been working towards creating more standards to support consistency, traceability, and reuse of results data. And I think, like everybody else who's a panelist on this call, we're really excited to see how our work can support this really important FDA initiative. Next slide, please. So I just want to do a little bit of level setting for our current analysis results standards for CDISC, what we have currently. And, so on the right, you see an analysis ready Atom data set and kind of below a static display. Unfortunately, we don't have a lot after the Atom data set right now to document results or metadata. And so we have static results that are created for the clinical study report. This can result in hundreds of tables and PDFs that are really hard to navigate. And there's variability between sponsors. We've even seen in our project, like, are you going to indent once or twice? Things that, again, don't impact the results, but then you see that variation. And it's expensive to generate these when you're just putting them in the static display. So there's not a lot of reusability. Next slide, please. So I do want to mention that we do have ARM analysis results metadata for define XML as kind of a limited way to currently document metadata. This is done usually after the fact. So you're going from your Atom data set right to your display, but to provide some limited traceability, you're providing information on the table names, basically helping the reviewer to, try to provide some traceability back to the Atom data set. Next slide, please. But again, ARM really describes metadata about displays at a high level. We don't have a formal analysis and results model. We can't use this metadata as is to drive automation. There's been limited regulatory use cases. So when people submit, it's usually for the primary or secondary analysis, but there's been kind of limited uptake by regulators. And again, there's this limited traceability. Next slide, please. So really, when CDISC is approaching analysis results, we want to shift to the way that we're thinking. So now we're starting with the Atom data set. Next slide. And we want to use the metadata that we already know is predefined, that exists, like we've seen already in the displays that have been shown. Right now, we're working on an ARM technical specification. So this is making sure that all of the metadata is in place. And this can be used along with the Atom data set, next slide, to really help with automation. So driving both a display, but also an analysis results data set, which is machine readable. If you think about the display, it's taking all of that information out, minus maybe some of the formatting, and can put it into a table for reuse and traceability. And I'll show an example of that in a minute. Next slide. So again, we really want to, going forward, we want a formal model for describing analyses and results as data. We want to help sponsors with automation. And we want to go from kind of the static displays to machine readable results to improve that navigation and reusability. We think this is really helpful in terms of supporting storage and access, as well as reproducibility of the results. And very important is providing that traceability. That's something that we have heard time and again, that it can be challenging to understand how to get back to the Atom data set. And finally, open source tools to design, build and generate analysis results. I think some people are thinking, wow, this is a lot of metadata. What do we do with it? And obviously, if you can have tools to help you process that metadata, that's helpful. Next slide, please. So this is our team's goals. So in terms of, we have the analysis results technical specification that I mentioned to help drive automation, the analysis results data set to support reuse. And we've been using common safety displays, so machine readable common safety displays to help set kind of this example of how it would look. And we've leaned heavily on FHUSE. Next slide, please. So quickly, this is just from a FHUSE white paper, just highlighting that you can really break down key metadata elements of a table and kind of label them. Next slide, please. You can also do this, again, this is right from the integrated guide. You can do this as well with the metadata from the integrated guide. You can kind of break it out and label it. And next slide. And you can represent this in a data set. So this is just a very brief and oversimplified example. It's not all of the metadata that would go into the table, but next slide. You can kind of see where you would have the result statistic and how you might be able to trace that back much more easily. So you know what data set it came from, you know the variables used, you know the submission value. So really traceability to the underlying data set. Next slide, please. We also want to develop a schema for machine readable TFLs. So this is something that we've been working on. How do you provide sponsors with that metadata right up front? We want standards to be easier to use. That's one of the goals at CDISC. And so providing that metadata to sponsors already in this format, we think will be very helpful. Next slide, please. And again, just thinking about what is our end goal, and it's really reducing unnecessary variability. So if you're thinking about collection, tabulation, analysis, all the way through the TFL, you can see that if you understand what the end is, you can kind of go back and provide that standardized metadata. Next slide, please. So then if you're going forward, you can start with the C-, think about what you're collecting and what you need to support in terms of analysis results and your TFL. Next slide. And this is my last slide. I know we're short on time, but I think CDISC really wants to voice support for this really important initiative. So what we're planning to do is take a selection of the FDA tables and figures and creating packages. So the machine readable displays, the associated analysis results metadata, examples of the underlying ATOM data sets, and make those available on the CDISC website, as well as creating that schema for TFL shells. And so really helping our end users and understand how to kind of implement what FDA is looking for. Thank you very much.
Okay, thank you, Bess. Now, next we will hear from Jeremy Wildfire.
Hi, everybody. Thanks very much for having me on this great session. My name is Jeremy Wildfire, and I'm presenting on behalf of the Interactive Safety Graphics Group. And I'll be talking a little bit about innovative approaches to safety analytics. Next slide. So the Interactive Safety Graphics team is a part of the same ASA BioPharm DIA effort that Barbara presented earlier. The ASAP work sits here as well. So the Interactive Safety Graphics team is focused on creating open source graphics for monitoring clinical trial safety. Big focus on a collaborative multidisciplinary approach, which can be seen kind of by the collaboration between ASA and DIA at the heart of this effort. And we're always looking for new team members. So if you're interested, I've included a link to let us know and we can chat. Next slide. I've included a few of our deliverables here. Our primary deliverable is an R package called Safety Graphics. It's available on Crayon, all open source, linked to a demo here as well. And I've included links to a few papers. 2 of the papers are from our team, and 1 is from an external group who's used some of the tools in support of the DMC. So yeah, next slide. There's some of the guiding principles of our team. So everything we do is open source, highly collaborative. I'll talk more about that in a second. We tend to focus on interactive graphics, but not exclusively, and I'll talk more about that a little bit later. We want everything to be very easy to use. In general, you just need a web browser to use our tools. Everything is both CDISC compliant and uses an extensible data model. So our goal is usually to support both that CSV you get from the lab on day 1 and the ATOM data that you have on day 1000. So I'll talk a little bit more about how that works. Everything we do is agile, and we really try to engage our end users as part of our collaborative approach. Next slide. In terms of collaboration, lots of different organizations involved across different biotech sectors. We do have a public-private partnership with CEDAR, so we get great input from our colleagues at FDA. Lots of work across functional areas. So all of our working groups have both practicing clinicians and analysts, data scientists and statisticians who collaborate. We also tend to be technology agnostic as much as we can. So though we have a core R package, there's lots of JavaScript code and other things involved. We all use SAS at times as well. So try to really take a flexible and forward-looking approach for how we build our tools. Next slide. Just talking a little bit more about the data, our approach to data structure. So this ties in nicely with the model we were looking at in the last presentation from Bess. So we tend to have 4 different components of the charts we make, and that includes study data, as well as a data mapping that identifies the key columns and fields in your data, along with some general inputs that can be used across lots of different studies. So we do try to have chart specifications and chart mappings, which map to different components of Bess's last few slides. So we've had kind of an exploratory implementation of the model that Bess was describing, that we're using in practice in this R package that we've developed. Next slide. So all of these components come together to make, in this R package, to make a shiny web application. So analysts typically will initialize these tools in R, and then the end users typically just go to a web browser and look at a hosted version of this application. So you can either view this in your web browser, kind of as a full-blown application with some interactivity built in, but we also support exporting chart code and standalone reports for everything I'm going to show you in the upcoming examples. Next slide. I think that, so we have several, we have lots of different charts that we've included as part of this framework that we've created. And I was very happy to see when I looked through the integrated guide in the last few weeks, there's lots of overlap. So the next few slides just kind of show side by side some of the tools from ISG that are available in this open source R package and some of the displays from the integrated guide. Not all the formatting is identical, but an awful lot of the content is similar. So here you see a system organ class level display of adverse events. The next slide, please. Here we drill down to look at here so the chart on the left is by FNQ. Our chart's by system organ class and preferred term, but all of our data mappings are very flexible. So if you have a data set that has FNQ instead of preferred term, you can just as easily display that in the chart on the right. There's also some of the functionality in the charts can be accomplished very easily with some of our interactive functionality. So you can filter by prevalence, you can drill down from higher level to lower level terms. Next slide. I'll just flip through these quickly. This one looks at EDISH, so very similar. Our chart is paired with a very detailed, it says 8, it should say 80-page clinical workflow. Next slide. Results over time, very similar. Next slide. And then finally, the shift plots, and the integrated guide does look at both min and max values in the shift plot, and we support that as well. Next slide. To wrap up, our group, we're excited that we're already fairly aligned with this integrated guide and think that we can align even further. So we're hoping to update some of the default configurations and also work on automatically generating some static charts to go with the exploratory interactive displays that we already have. And several of our working groups focus on domains that are included in the guide as well. So, and that's all from me. Thank you.
Thank you, Jeremy. So thanks to all the panelists for those opening remarks and presentations. So on the next slide, we have a few questions to get us started. And before we get into these questions, I wanted to ask, are there any clarifying questions that the panelists have for the presenters?
Yeah, so one quick question. So sometimes throughout the FDA presentations, we hear things like, for example, right, if the clinical reviewer wanted percent change for labs, they can request it. If then when that happens, who are they requesting that of? Or when those comments are made, is that more towards they might request it of the FDA statistical programmers, or are you thinking they might request it of the applicant?
So it can be both, but let me just say that there are also a few questions in the question answered and chat related to this. So one of the question is that when will FDA start requiring this? Well, I want to make sure and clarify that we are sharing this in the spirit of transparency. So internally, we do have a clinical data science team, and they are providing these standard safety reports to the reviewers for the applications that are under integrated assessment, and a few more, meaning, some important efficacy supplements or new molecular entity, some of those are also served by this clinical data science team. But we also consider that for the applications where there is no, this clinical data science analyst support, reviewers do ask the sponsors as well if there is a clarification needed or if the analysis needs to be changed. We also have statistician colleagues who help out with the safety analysis as well. So there are several ways the reviewers, clinical reviewers, can request this analysis. In general, we are not making the integrated guide a required analysis for the sponsors to submit in the NDA applications. If they do, it will be very helpful because then we are looking at the same analysis that the sponsor is looking at it as well, but there is no requirement at this time. All right, so now, there are a couple of clarifying questions. So before we get into our panel discussion, maybe we can just answer those with the help of panelists. So one of the question is, do you take the CTCAE grades for lab data in defining the cutoff for the outlier analysis for the lab data? And table 59 doesn't take into consideration of baseline values. What if the patients have abnormal values? How is it addressed? So I would say here that when this cutoff table was designed, several existing grades, like CTCAE or HIV grading system, healthy volunteer grading system, all of those were taken into consideration. So if there is an oncology application, the NCI CTCAE grades are completely fine. So that can be used as well. So we are not saying that only the cutoff presented in the table 59 should be used. If appropriate for the application, please discuss with your review division, but whatever is that therapeutic area is using is completely fine. All right, so we will now move into some questions for the panelists. And one of the questions is, what are the strengths of the integrated guide and how can it be improved? Especially that this is sort of a first time we are making available our analysis that we do for safety to the public domain. So we would love to hear your reactions to the integrated guide. So we'll start with Mary.
Yeah, so I think the main strength of the guide is really just it's always appreciated when there's transparency, right? It's always good to know what our main customers, being our clinical reviewers, find helpful and what you are doing with the data that we provide you. So the transparency is always appreciated. As far as areas for improvement, I think the one thing that our FUSE group can do when we provide comments is to provide suggestions for additional references, right? To provide references for supplementary type educational information. I think that would be very helpful to again, kind of expand on that educational concept, because we think that's important for whenever you have standards. Because if people just blindly follow standards without having the appropriate expertise to know if and when to use which standard, we think that's important. So we'll probably help with suggesting some references to help with the education piece.
Great, thank you. Bess?
Sure, I mean, I think just to echo what Mary said, I think people always appreciate that guidance and transparency. So we don't have to have maybe those conversations around whether things are bold or what kind of font they're in. And I think especially it gives us a really great starting point, as well as insight into what kind of the agency needs in terms of safety reporting. In terms of improvements, I think really, I think like Mary, there's really an opportunity for collaboration. I think one thing that was presented earlier was around the NASH tech spec. And I know in the tech specs, they kind of go into greater detail about maybe possible data sets. We would be very much interested in collaborating and trying to maybe establish that ATOM metadata so that there's really good traceability from the results back to the ATOM metadata, just because you have everybody's giving you the same output doesn't mean that underlying data is the same. And so I think that's important. So I think it's exciting and there's some really good collaboration opportunities.
Thank you. And Jeremy?
Yeah, I think the integrative guide provides a useful set of chart specifications, right? I mean, there were no big surprises in it for me. But I think right now everybody's deriving these charts with their own set of SAS macros in 100 different ways. So I hope that having this shared set of specification will lead to more standardization and hopefully to a more automated process for generating all of these things. And that kind of goes back to some of the data model things that Bess and I both talked about a bit in our presentation. For the limitation, I mean, I would imagine creating this full set of tables and figures would result in hundreds or even thousands of pages of figures to review. Even though when you look at it, there's really maybe only a few dozen templates of the different things, but you're creating so many duplicates. So I really think that's one of the places where some exploratory tools that both Mary and I have worked on can really help. So instead of creating 1,000-page PDF, you create an interactive tool to go with each, or even create both, and you can start with the exploratory piece and then really look at static versions as well. And if you can automate a lot of that, then I think everybody will be in better shape. So.
Thank you. How about, can you share your thoughts on the treatment emergent AE approaches that we discussed earlier? And this time, we will start again with Mary.
Yeah, but there's, treatment emergent has been one of those topics that always sounds so simple, but it's just not, right? So it always seems simple. Well, it's those that first occur or worsen after initiation of treatment, and it always sounds so simple. So the devil is in the details. So I think the definitions you provide in the slides is certainly helpful. I think, moving toward this on study on treatment type thinking is consistent with the S-demand framework, right? So the efficacy people, are very much getting into the estimate framework, and for safety, it's just a matter of time before we start thinking in the estimate framework as well. So it's very similar to the treatment policy estimate and the on-treatment estimate. So it really is good to start thinking in that direction. Now, the definitions on the slide, I think would work very well for a very simple study design, right? So a controlled treatment period design followed by a no drug follow-up period. Where things get really tricky is when that's not your design, right? If you have a more complex design, like so for us, it's not very unusual to have a treatment period followed by an open label extension period followed by a no drug follow-up. And in that case, having no cutoff for the on-study estimate isn't going to work, right? You're going to have a cutoff. And similarly with the on-treatment estimate, adding days, same thing. The study design matters, right? So if you have an open label extension period, it may not make sense to add days based on the half-life of the drug because you're going to introduce bias by, there's no way you're going to attribute the open label extension data toward placebo, right? So you're going to have cutoffs, and sometimes it just doesn't work to have a cutoff based on the half-life of the drug. So study design matters. And then also, it was already brought up in your slides about rescue medication. Rescue medication matters, right? So it may not make sense to add days based on the half-life of a drug if you've had the introduction of a rescue medication. Again, you might be attributing the events of the rescue medication toward placebo, right? And you can miss signals. So I think we share the passion to try to reduce the variability of how we all define treatment emergent. It's just harder than it seems. So yeah.
For those insightful comments. And Jeremy?
Yeah, I mean, I agree with much of what Mary said. Like having guidance is helpful, but science is hard, and there's going to be variations between studies. I thought that the definitions were a reasonable starting point for sure. It also made me think a little bit of Dr. Unger's comment about narrow versus wide FMQs and how 95% of the time it doesn't lead to a different interpretation, right? And I expect that's true here as well, right? So having that exploratory layer where you can quickly toggle between on study versus on treatment, and even change the follow-up time with a little slider and see if the signal changes can be really valuable instead of having to generate dozens of displays for the different iterations. So it's another point where pairing the exploratory with the formal final analysis can help do that sanity check in an easier way than we've done in the past, maybe.
Great, thank you. I also like to mention that, this was noted earlier that, many organizations, they have their own macros for tables and figures. And I wonder if your institution has that, and if it is, then what were your lessons learned, or what obstacles did you have to overcome to develop, such an effort to create a standardized set of analysis results? And in this one, I would say, if we can start with Bess.
Sure, and I can just speak to our group in terms of trying to think about the most common safety displays. So kind of gathering them up from kind of various sponsors, as well as the FUSE group, and we spent quite a long time trying to come to again agreement as well, and I think what we have is very close to FUSE, and I think that's also very close, what you presented. But yeah, I mean, I think having that, that common ask around, the type of metadata that should be presented will be incredibly helpful to reduce that variation between, do I want this horizontal or vertical, or all of those kind of conversations.
Thank you. Mary?
Yeah, so yes, in my organization, we do have standards in place for both, for the data and also for the standard safety analyses. And at my company, we have very much aligned with the FHUSE white papers. And so we have certainly leveraged that material to get to our standard tables and figures. And certainly that it's still challenging since we're involved in lots of therapeutic areas, and it is hard to always get alignment, but having the FHUSE white papers have really helped because that just turns into our default place to look. So we do try to maintain. So yeah, it is a difficult thing to maintain, and certainly with the new guide and possible updates, we'll be working over time to make any necessary updates. So it is a constant maintenance challenge.
Thank you, Mary. Jeremy?
Yeah, the macros at my organization predate me by probably a decade or more. So they've been around for a very long time and have changed constantly. I will say it's been interesting working in the open source world, in the cross-functional, like the cross-company space, and seeing more and more collaboration there, because things really are, like, the implementations may be different and the code is certainly different, but I think the core outputs are quite similar. So I'm very encouraged to see more and more companies sharing code. I think it's still early days for sharing safety visualization, safety TLF code, but there's more and more code to generate ATOM data, for example. There's a big new R package that's a multi-company collaboration to derive ATOM, which I think is pretty incredible. So I think more and more, hopefully there will be shared code across companies instead of these monolithic macro libraries that we all spend years and decades maintaining and updating.
Thank you, Jeremy. One more question, and I think this is right, up on your alley, Jeremy. So what new approaches or technologies or methods can enhance identification of pre-market safety signals in clinical trial?
Yeah, I mean, we've touched on it a little bit already. I think that really what I hope to see is the ability to pair these exploratory charts that we've been talking about with the fully validated displays that are in the integrated guide, and to really have them seamlessly appear in the same place, in the same analytics framework. So I think that the foundation to do that is largely built on Bess's team's work at CDISC, and that paired with some technological improvements in our industry, now that we're moving away from storing everything in folders on a file server and putting things into databases, clinical data repositories with clinical metadata repositories and statistical compute environments. Those things are becoming much more possible as we kind of modernize our tech stack and pair that with the robust data standards. And then I think Bess did a really good job identifying some of the gaps in the chart specifications and things like that, that is really part of the last piece of the puzzle. And then things start getting really interesting when you talk about truly modern statistical methods and AI techniques, which I know FDA has released a bunch of guidances on in the last few years. But I feel like the foundation to do some truly innovative technology in our space is in place now, when maybe it definitely wasn't 10 years ago or even 5 years ago. So I think we're trending in a really good direction there.
Thank you. Bess?
Yeah, I think having those robust standards in place, and I was thinking of this when I was looking at the kind of the entire list of questions, but also how do you put conformance rules in place around the analysis results metadata, and making sure that kind of people are able to conform. Obviously, we have conformance rules for our other standards, but yes, I find that would be supportive.
Mm-hmm. Okay, great, thank you. And Mary?
Yeah, I think as far as new approaches and technologies, I do think definitely moving toward interactive methods is the way to go. And I'll also agree that we need to do a better job sharing, right? So I think the vision of what was, the safety analytics team, or previously known as code sharing, right? There was very much a code sharing portion of this. And so hopefully in the future, we're all creating shared interactive tools. We're all sharing them and using them. And in the early days of the FDA PHUSE collaboration, the buzzword was crowdsourcing, right? How do you crowdsource the development of these things so that we're not all individual companies aren't making their own, and each regulatory agencies making their own? How do we create these and crowdsource them? So I think that's where hopefully in the future, we're doing more and more of that and developing really innovative interactive tools and create a very nice library. As far as statistical methods, I think there's a lot of room for advanced thinking, right? We do very much rely on the percentage and exposure adjusted incidence rates. There is likely other statistical tools in the toolbox to maybe more widely use, and some of which Matt already alluded to. So maybe some of our time to event-based approaches might end up being better, because the exposure adjusted incidence rates do suffer from that constant rate ratio type, the constant risk over time, which we don't always have. And we're well aware of some of the limitations of percentage. So I do think there's room for innovation with statistical methodology as well.
Thank you. I like to also just, answer one of the question that may be sort of brewing here, is that, what about the interactive graphics, and I would say that right now, internally, we do have tools to really help us drill down into the data points to the subject level data from the high level summary statistics. So we are also internally using both the static and the interactive visualizations. And the topic of whether or not FDA will accept it is sort of beyond the scope right now. Here, I see some of that in the questions coming up. But in future, once we have some of the initial discussions taken care of, then, it's something that we can discuss further. There are some questions in the, question answer chat. One of the question is, what are the panelists' thoughts for sponsors on mapping to this framework during the development of the investigational product, starting with the end in mind? So any reactions to that question from the audience? So Mary, do you want to go next?
Yeah, I want to make sure I understand the question, whether or not we should be creating our tables and figures consistent with this guide in our integrated summary of safety and summary clinical safety.
Right.
Yes, I think all of us in industry is going to be struggling with that. and I think it is a very important question. And in my mind, it, hopefully we'll over the next week or two learn more from the FDA on timing. For example, I know you're gathering comments by October 31st. And do you think you're going to have an updated guide anytime soon after that? And if so, some of us may want to wait a little bit. And some of us may want to wait until there's some ADAM specifications in place before we go crazy on updating our standard code, right? Because it takes a lot of time to update our standard code. So we don't want to update it, like urgently update it now, only to then, oh, well, we assume different ADAM structure. And now there's a new guide out, an updated guide. So I think this is a difficult question for those of us who have to make these decisions. Hopefully we'll learn more in the next week or two. And then, I'm sure all of us will be trying to figure that out.
Thank you. Jeremy?
Yeah, I mean, I agree with Mary once again. I guess I would say what's been published now might be a good time to do some baseline analysis and gap analysis of what your standard safety submission looks like. And if there are big chunks of the guide that are not covered at all, maybe add them in, but don't worry about the formatting just yet, So I think it's really helpful to see the approach that's used internally so that we can start aligning now, and then we'll see how everything evolves.
Great, thank you.
And maybe just to follow on comments with that, obviously, when you're in the middle of something, it's hard to change. But I think in terms of being able to create some of that metadata kind of further guidance, especially if we can kind of take that all the way back to collections. So for new studies that you're thinking about, because it's always hard, I think, to do something kind of like when you're in the middle or try to retrospectively fit it, and where you really see that. I think that value is when you're kind of going forward. So how do we, work towards that in terms of, again, starting with the end in mind, but making that kind of defining that up front as well.
Thank you, very helpful. Now, there are a couple of questions for which I'd like to invite Dr. Veronica Pei for answer. So one of the question is, can you comment on the use of IDISH plot in a population with liver disease? And the second question is, when can FDA require manufacturers to submit DILI data for NASH technical guidance?
Okay, so I think I answered the second question in the written comment. Currently, every application that is submitted to the FDA for new market applications, the requirement is that they should have a DILI analysis. However, up until now, really, we have not really provided any guidance as far as data submission, and so the intent of this guidance that we published for NASH is really to help with that. But that is not a binding at this moment. It's all the FDA guidances are not required. It's really highly recommended that sponsors should discuss with the review division and really look at the recommendations. And again, I think when we developed the guidance, we left it very flexible so that there's lots of considerations and lots of recommendations for the data specifications, but it's very flexible and really is dependent on each indication and the underlying population. As far as for the population with liver disease, and I think, it's still debatable right now, even within our own SMEs, what cutoffs that we are using to assess specifically for potential DILI. And so what we presented, the default cutoffs were for, in general, for patients with normal underlying liver function. And we think that, in patients with underlying liver disease, they're not necessarily at increased risk for DILI, but when presented with, when they experience a liver injury, perhaps they're at more risk for more severe liver injury. So the question is then, what cutoffs should be used? And again, I think that should be discussed with the review division, specifically, for drug products that do have potential DILI as already suspected through preclinical studies or in phase 2 studies. Then in general, we do recommend that the sponsor discuss with the review division early on, and they can also consult the DILI team to reach agreement on the specific cutoffs to use for the analysis. So I think the FDA is very open to proposals from the sponsor to kind of reach agreement on those.
Thank you, Veronica. And the last question here we have is, what about shift tables for lab parameters? And what I would say here is that we do use them when it makes sense clinically. So for example, if you have patients moving from pre-hypertension to hypertension, from hypertension to hypertensive urgency or emergency, those clinically meaningful brackets, it really helps to have those shift tables. So we have considered them, and they are part of the follow-on guide. So I know we are running out of time now. There are lots of other questions. We would invite everyone to sort of send those questions to the docket so we can answer those questions later on as well. But thank you, everyone. And back to Marianne for closeout of our workshop.
All right, thank you so much. Thank all of the panelists. So for just a few closing remarks, the FMQs and the Standard Tables and Figures Integrated Guide represent efforts to enhance pre-market safety evaluation of submitted clinical trial safety data. These efforts by the FDA are complementary to ongoing initiatives taken on by other multidisciplinary stakeholders and organizations leading the safety assessment ecosystem forward. As you've heard throughout the day, FDA welcomes your feedback on both the FMQs and integrated guide and continuing the conversation around pre-market safety analytics. We were just very happy to see that was such a volume of audience submitted questions and such a lively panel conversation. And there's so much interest in the development occurring in this space. So one more time, we'll plug. I want to encourage all of you, both in the audience and on the panels, to contribute your comments to the docket that FDA has established to receive input from stakeholders. The docket will be open until October 31st, and the link can be found in the chat. It can also be found on the workshop event page. So with our last few minutes, before you go, I'd like to take a moment to thank everyone who contributed to this excellent workshop. First, all of the speakers and panelists who continue to do great work in this space and who took the time to share their expertise with us over the course of this workshop. Thank you as well to our colleagues on FDA's planning team for this meeting, including Fashali Popat, Scott Prestel, Veronica Pay, Nee Beasley, Christopher Smith, Douglas Warfield, and Matt Sukup, along with many more at FDA who contributed to the planning process. And then finally, a big thank you to our team here at Duke Margolis, including Brian Cantor, Duray Kim, Nancy Allen Lapointe, Thomas Rhodes, Victoria, Jim, Derek Rapista, and Matt D'Ambrosio. Finally, I'd like to thank all of you in the audience. Thank you for joining us for this workshop and have a great rest of your day.